All right, we're back with part two of the DNA technology. Um, we are where I left off was talking about this idea of a cDNA library. These are really cool ideas because remember, if we look at our cells, every single cell in our body has the exact same set of DNA because it's all us. But what makes those cells differently is what RNA they're actually using, which changes as you know things change in your body too. It's not always the same, so it's not a constant thing. So what we do here is we have our cell as the DNA inside the nucleus and the RNA. What we do is we actually destroy the DNA and we destroy the rest of the cell, and what we're left with is those RNA. We take that RNA, we steal a little tool from viruses. With the HIV has this thing called reverse transcriptase that actually takes the RNA and it and transcribes it back into DNA. And then we use that DNA as a library and create what is going on in that cell at that time. It's a very cool idea, and we'll use it a little bit later on as well. Um, so there are a few differences. So we can't just take a human gene and put it directly into a prokaryotic organism. So if you remember, there's this little step in gene to protein where in eukaryotic organisms, they actually edit their DNA. Bacteria don't have that ability to edit. And so what we need to do is make sure that before we insert a gene into a bacteria, we need to edit it before it goes in there. We have to cut out the introns. And we can do that a number of ways, one of which is the way that I just described, that idea of the cDNA, using these ability to take the RNA that's already been edited and turn it back into DNA and then insert it into a bacteria. Um, they have these things called yeast artificial chromosomes. They're big. And we can also take DNA back into that to eukaryotic organisms by this using an electric shock essentially to put it in there. All right, organizing DNA. This is a big one, gel electrophoresis. It's this process that's chromatography. It's a fancy name for getting things to separate. And it's a pretty simple idea, but it looks much more complicated. What we do is we cut up some DNA, and when we cut it up, it's in lots of different fragments, but those fragments are different sizes. So what we can do is actually sort it by size. What we do is we put a few drops of this DNA that's all mixed together, all these different sizes, and we put it into a gel. And when I say gel, it literally looks like Jello. Um, and we then put an electrical current there, which actually starts to move the DNA. DNA has a positive charge. Has a negative charge, and it moves towards the positive terminal. So if we put an electrical current there, the DNA will start to migrate, and it migrates through the gel. And depending on the size of the DNA, it can move at different rates. Small pieces find it easy to sort of wind their way through the gel, and so they move very quickly, and large pieces move very slowly. And they do this at a very consistent rate. So if you turn this on for a little while, let it run, and then turn it off, what you'll see is a pattern, and we'll see it down here. So this is where we load our DNA. The DNA this thing gets turned on, and the DNA starts to migrate through this. And then what we see is that the DNA fragments that are small end up on the edge here, and the DNA fragments that are really big don't move that much. And in this way, we've actually separated the DNA, and we now know what the just so you know, this is not just one piece of DNA. This is, thousands or hundreds of thousands of the same size piece of DNA. So there's an example. So just so you know, this is also one of the earliest ways in which we did DNA fingerprints, that you would take DNA from a crime scene and DNA from a suspect, and you would cut them up and run them by each other. And if they come up with the same banding pattern, then they're the same person. If you ever catch me and have some time to kill, I'll tell you the whole saga about O.J. Simpson and how he got off, even though there were DNA fingerprints that showed that not only was his blood at the crime scene, but the victim's blood was in his car. So you tell me how that happened. Anyway, polymerase chain reaction, another really exciting. This is tons of words, and I'm going to sort of skip over it. But this was an unbelievably revolutionary idea here. Up until the mid 90s, the only way that we could copy DNA is by putting DNA into a cell and letting the cell replicate the DNA. It seems logical, but it was complicated. So there was this idea like, well, could we do it just in a test tube? And they started to do that. And what they did is they used some very cool techniques. You wrote that essay on DNA replication. You know that there's a lot of enzymes necessary, but we can actually take a few enzymes out of it if we do some tricks. For one, we don't need DNA helicase if we just apply heat, because heat will actually split the DNA apart. Those little hydrogen bonds that hold together the A's and the T's and the G's and the C's will actually be disrupted if we just heat up the DNA a little bit. So that's one of the techniques here is that we of course we need DNA polymerase. There's no way around that, and we have to get that to work. And we also need primers. So that's another thing. We need some sort of primers. And the cool part about this is that once you learn how to do this PCR technique, you can actually target very specific areas of the DNA by changing the primer. But I'm going to show you a video because I think it will be helpful. This is the idea here is what we do is we take the target sequence, we heat it up and split apart the DNA, cool it down a little bit so that these primers add on. Okay, and then we let DNA polymerase start extending the DNA, and we do this, we cycle this over and over, heat up, cool down, heat up, cool down, and when we do that, we actually every cycle we can theoretically double the DNA. This is an example of this little video, and hopefully this will help out here. There's our DNA, and you can see the thermometer on the left there rising. We're going to take it all the way up to 95 degrees Celsius. That's very hot, and so what's going to happen is we're going to split apart this DNA down. We're going to unzip the DNA down the middle, okay? And that heat does that. And then what we're going to do is we're going to cool it down to 55 degrees, which allows these primers to come in. And there's an upstream primer and a downstream primer, because this is the target sequence right in there. And then we're going to heat it back up to 72 degrees, which is what the DNA polymerase is going to need to start adding DNA, and it's going to extend this out and copy the DNA. Notice we still didn't get the piece that we want. We only want this little piece, but we're going to cycle it again. And we're going to bring in we denature it by heating it up, we're cooling it down, we're adding our primers, and then we're going to do this. And notice we still don't have the fragments that we want, but we're going to do it again. And now you're going to see that we're going to start to get the fragments that we want. And these are our target sequences right there. Boom, boom. That's what we want. And those guys are going to become more and more numerous as these cycles go on. Typically, you run this for at least 20 cycles, maybe 30 cycles. And notice we have lots more target DNA as we go. And the cool idea is this: we can take one molecule of DNA and turn it into over 1 billion molecules in 3 hours or less. It is really incredible, and you don't have to insert it into any living organism. Okay, another helpful hint is this idea of DNA sequencing. Basically, this is PCR with a twist. So what we're going to do is we're actually going to replicate the DNA, and instead of replicating it into an entire target strand, what we do is we replicate fragments, and we end them with these fluorescently tagged DNA. Whenever these fluorescently tagged nucleotides get on, it stops. And notice that they don't extend. Once you actually get one of these colored coded ones on there, they don't grow anymore. And what it allows is you to get different sized molecules, each with a different color coded tag on there. These are randomly produced, but here they're shown in order from shortest to the longest. But these would all be mixed together in a test tube. And what we do is we run it through a gel electrophoresis. And the small molecules are going to move fast. That pink one is the fastest moving. Then this green one is the second fastest moving. And they will move past a detector, and you can actually detect each nucleotide. These machines that do this, they're about, $1 million and a half, but within a couple hours, they can sequence over 1 million bases and figure out what your DNA codes is. It's no longer that difficult. One of the surprising things is we found that we were not expecting humans to be a small number of genes, which they are. I mean, we're still bigger than bacteria and a lot of simpler organisms, but there's plenty of organisms. There's fish that have more genes than us. Rice has way more genes than us. So it's really one of these things that was a surprise to us. We were expecting humans to have more DNA than everybody else, and we don't. So the complexity of an organism is not directly related to the number of genes. Last technique, microarrays. They're really, really amazing. Microarrays, I'm going to skip right to this idea here. What we're going to do is we're going to take our sample tissue, okay? We're going to take our RNA. This is just the genes that are turned on, and we're going to turn them into DNA, but we're going to turn them into glowy DNA. And they're single-stranded little pieces of DNA. And so they have their bases are all out there to see. And what we do is they make this, and this is really the remarkable part, this little silicone microchip here, and etched into the glass on each of these dots is a single gene. And these genes match, or are they have the opposite letters as some of these genes. So what happens is when we put this glowy DNA onto this microchip, these molecules start to slide around on here, move back and forth until they find their match. And when they find their match, they glue themselves down because of that base pairing rule. And what we can do is now we can see which genes are turned on. They literally glow. Each one of these dots represents a different gene in an organism. It's really amazing because what it can do is this technique allows you to sort of tell the difference, for example, between a cancerous cell and a healthy cell. What genes are turned on in that cell and what genes are turned off. And it can tell you really quickly the difference between those genes. And this technique has been really revolutionizing how we treat cancer. Stem cells, we'll go to Dolly right away. Dolly is the sheep here that we first cloned. We took a DNA from this is the mammary cell. We took the nucleus out. Then we took the nucleus from another sheep, put it in there. So this sheep down at the bottom, Dolly, is the identical clone of this sheep up here. And that's cloning. Can we do it in humans? Of course we can. Is it ethical to do that? That's a whole different story, and I don't know if I can truly answer it. I will tell you, this sort of cloning doesn't serve a lot of purpose because it doesn't help you that much in the end with what is to treat anybody. We're just making an identical clone. Maybe if you're trying to make the greatest basketball team ever and you want to clone Wilt Chamberlain and Michael Jordan and LeBron James all at the same time to be all in their prime, that would be an awesome idea. But other than those sorts of silly ideas, there's not a lot of medical uses for this. They do this in animals. You can get your pets cloned. There's a company in California that you can send your dog DNA out there and they will clone it for you. Again, you don't get your exact dog, you just get a clone of your dog whenever you want it. So kind of interesting. It's very expensive, by the way, so I would not recommend it. All right, we're gonna have one last video. It'll be short. I apologize.